Hi, welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. Folks, today's examined content is going to be further dissecting the paper that we discussed in the last piece of content. So this is part 2 of that examined piece. In this examined piece, we're going to be covering how fasting mimicking dieting can affect cancer, stem cells, and immunology. So without further ado, let's jump into this examined piece. So to introduce this particular part of the paper, we're going to be looking at fasting and fasting mimicking diets, impact on cancer, the effect on the immune cells, and certainly also stem cells. So a number of different markers, some really exciting stuff, and I'm really looking forward to getting into this. So without further ado, let's jump into the experimental methods so we can understand those a little bit better. So in this section, we're not just going to be looking at mice, we are also going to be looking at a little bit of human data on a pilot study that they did for FMD, fast mimicking diet, and its impact on stem cells, particular stem cells. But in terms of the cancer and in terms of the immunology, they looked at mice markers because they have to sacrifice the mice, they have to kill the mice to be able to get certain biopsies and just to get certain muscle groups and things of that nature, essentially just harvesting different parts of the animal. I realize that's a bit gruesome, but unfortunately that is part of science when you want to get into the nitty-gritty of the molecular mechanisms, the cellular aspects. So again, the mice went through an FMD diet essentially for a large portion of their life until they were elderly, and they did so for 4 days, and that was based on their chow, their particular food type that they consume. Now in terms of the humans, they did it for 5 days, and when they did it for 5 days, they did it for 5 days several times within a 3-month period compared to controls that did not go through the FMD diet, so they just ate normally. So with that out of the way, let's jump into the results of cancer, stem cells, and immunology. Let's jump into it. All right, the first figure we're going to be looking at is figure 1Q. Figure 1Q looks at a particular marker in satellite cells. Now, satellite cells are specific to musculature, and they're essentially kind of a quasi stem cell. And these stem cells, or these satellite cells, will integrate themselves into the musculature and donate their nuclei. So that's why they're not necessarily 100% considered a stem cell, but they are stem cell-like. And PAX7 is a marker of regeneration and survival of these particular satellite cells. And what's really fascinating here is that we see a decrease in PAX7, so that would be a negative outcome. That means you have less survival and less regeneration, presumably, of these satellite cells during the fasting phase, but after refeed, you see a rebound of this PAX7. So, a week of refeeding, you see this rebound of PAX7. Not only that, you don't not only see a rebound back up to normal basal levels for the same aged mice, you actually see it increase to the level of youth. So the same levels as adult, so younger mice, which is pretty astounding to think about. And we're going to have more data that's really interesting coming up. The next figure is looking at mesenchymal stem cells, figure 2T. Figure 2T shows that you see a decrease in mesenchymal stem cells with age. And interestingly, however, once you do this fasting phase and then this refeed phase, you see a significant increase in these mesenchymal stem cells. And you may be wondering what mesenchymal stem cells are, and that's a good question. Mesenchymal stem cells are, of course, stem cells, but they are slightly limited in the type of cells that they can turn into. The differentiating factor that makes a stem cell is the fact that it can turn, or there are a number of different differentiating factors, but one of them is that they can differentiate into another tissue or into another cell. They're essentially a blank slate that can allow them to turn into something else. Now, mesenchymal stem cells are called what's called multipotent. So they can turn into multiple different types of cells, multiple different types of tissue, but they can't turn into all of them. A cell that can turn into all of the different tissues that we have in our body would be called a totipotent stem cell. Now, mesenchymal stem cells can turn into adipocytes, so fat cells. They can turn into bone marrow. They can turn into chondrocytes, which are found in our cartilage. They can also turn into muscle cells. So they are certainly diverse, and they are absolutely necessary for our survival. But they aren't as powerful as a totipotent stem cell. But still, this is a fantastic marker of stem cells. So what we see, again, is this increase in these multipotent stem cells back up to their young levels of stem cells within these mice, which is pretty remarkable. So you're seeing essentially a regeneration, a repopulation of these stem cells back to not just their older controls, but actually beyond that to their younger controls. Now, the final figure I'd like to cover for the stem cell section of this content is a human pilot study. So yes, they did look at humans as well, and a pilot study is essentially where they just kind of interrogate the question, but they don't go full force. So they don't spend a ton of money on the subject, but they want to find out if some of these trends that are found in mice are also potentially found in humans. And it turns out that they looked at mesenchymal stem cells, which I just explained what those were. They're, again, these multipotent cells that can turn into a variety of different other tissues. And what they find with these mesenchymal stem cells in humans is that there is a trend. Now, this is a complicated topic because the p-value is set at 0.1, which is very close to that 0.05 marker. And they do mention in the paper maybe if they had had more subjects, which makes sense because, again, it's a pilot study, so it's going to be underpowered. There aren't going to be as many subjects or participants in the study as they might want. So if they repeat this procedure, maybe they would see a increase in these mesenchymal stem cells. I know that if you look at the graph, it shows this massive increase in the bar, but if you look at the actual error, the error is actually pretty large, and that's why they're seeing just a trend towards significance and not necessarily it showing that it's significant. So in this situation, I would probably caution against making any massive conclusions off of this data. So based on the data that's provided, I would say that the data is leaning towards fasting or fast mimicking dieting having an impact on stem cell regeneration and potentially more of these stem cells proliferating and growing, so the regrowth of the population of stem cells that exist. All right, now let's shift gears towards cancer. How does fasting impact cancer based on this paper? In figures 2I and 2J, we're looking at neoplasms. Neoplasms are a fancy term to say tumor. So I think you probably are familiar with the word tumor, so that's what we're going to use. And with fasting mimicking dieting, we see decreases in tumor size. So the actual tumors themselves are decreased from fasting. And if that's a general tumor or if that's a lymphoma, it doesn't seem to matter. In both conditions, you see decreases in the tumor, which is pretty remarkable. The next thing they wanted to look at was histology. So they wanted to look at a series of different cells, kind of if you have a flattened section, and if you were to just look at that flattened section, can you see changes in morphology? Can you see some sort of abnormality that you would normally see in something like a tumor? So I asked a friend of mine who is certainly more knowledgeable in this area than I am, and she did mention that there is greater infiltration of lymphocytes in this area, which would be indicative, or at least an indication, of lymphoma. So lymphocytes are a particular cell of the immune system, and if you have this rampant reproduction or production of these lymphocytes, then, well, that could be a sign of cancer. Not only that, though, you see that the circle that they placed on figure 2E, that there's abnormal mitosis as well. So mitosis is the cellular process by which our cells are able to, well, divide. That's what mitosis means. And in this situation, you see that there's an abnormal mitosis occurring in terms of you're not getting two separate cells. They're kind of stuck in a mitotic phase. So that is another signal of something that's irregular going on. Next, they wanted to look at when these neoplasms or tumors appeared on the animals. And really interestingly, what you see are that these neoplasms, these tumors, pretty distinctly show up later in life. So you tend to have a much healthier, cancer-free life for a much longer period of time, and then they present. So they present with older age, as opposed to the control animals, which had cancer essentially show up at any point in their life. The next thing that they showed is the number of lesions or abnormalities, tumors that would show up on the animals, and it shows again that the fasting condition had decreased numbers of tumors. So a fasting mimicking dieting mouse might have one tumor, while a control animal would have something like 3 or 4. So there are substantially more of these actual tumors. Not only are the size much smaller, but they also have lower number, and they tend to appear much later on in life. So really, really interesting right there as well. And for the final figure that I wanted to discuss, figure 5C on cancer, is you see this trend of much earlier deaths, and not only that, but much greater amount of deaths due to cancer in the control animals, but far less deaths from cancer and far later deaths from cancer in the fasting animals. So it seems like unequivocally fasting seems to have a significant impact on these animals in terms of cancer progression, cancer size, I mean all kinds of different measures, which I'll touch on in the discussion and conclusion section as well. So now let's focus on immunology. How does it change our immune system? Now, looking at immunology, we're going to look at two figures. So figure 2O and figure 2P. So figure 2O shows a white blood cell count. So white blood cells are essentially a general term for essentially immune cells. So you have an adaptive immune system and you have an innate immune system, which I'll touch on when I cover the next figure, 2P. But 2O essentially shows that you see a decrease in the number of white blood cells when you are fasting. And that makes some sense because we saw in the previous content that I released talking about organ size, you see atrophy. So it makes sense. So the entire system is essentially degrading. It's starting to lose size. It's starting to lose, body fat mass. From the previous section, looking at organs, we saw decreases there. Of course, we're going to see decreases. Well, I suppose I shouldn't say of course, but presumably we'd see decreases in the white blood cell count. So the immune system isn't going to be pumping out as many of these white blood cells. But interestingly, again, with the same thing happened with the organs. When we see this regeneration of these organs, we also see an increase in the white blood cell count after the fasting. So in the refeed condition, not only do we see white blood cells go back to their old cells, back to their old number, but it far surpasses it and goes all the way back to younger state cells. So they revert to a number that are comparable to younger mice. So the older mice essentially rejuvenate the number of white blood cells back to younger cells. That is really incredible stuff. The final figure that we're going to be covering is figure 2P. And figure 2P wants to look at the ratio in the immune cells. So these white blood cells, how many of them are made up of adaptive cells and how many of them are made up of innate cells? So innate cells are essentially cells that are just normally circulating through your system. So typically something like a macrophage, which is a large cell that goes around and goes through what's called phagocytosis. So it kills by consuming different bacteria and different pathogens and kind of broken materials that are found in our body. And those are normally circulating around. You want to have some of those just to kind of do the routine maintenance that your body needs. However, when you have an infection, if that's cancer, well, not that cancer is an infection, but if you have cancer or you have an infection or you have some other disease, you might see increases, and this is especially true for an infection or an autoimmune disease, where you see this massive rise in what's called an adaptive response. So these adaptive immune cells are going to essentially target a particular pathogen or a particular antigen and start to proliferate. So they start to multiply multitudes over, hundreds of times over, to a point where they completely overwhelm the system. So they're all over and they're just ravaging the entire system, trying to find this one pathogen that they're dying to kill. So in that situation, that would usually imply that there's something wrong, that your body's trying to get rid of something incredibly specific. So they wanted to look at this ratio. So in figure 2P, what you're seeing is that with the young mice, you have this high proportion of lymphoid cells, and lymphoid cells are part of the adaptive immune system. And then for the myeloid cells, or at least compared to the myeloid cells, which are part of the innate immune system, so I mentioned a macrophage earlier, that would be part of that myeloid lineage, that innate immune system. So the younger mice tend to have a higher proportion of lymphoid cells to myeloid cells. But as you see with the aging mice, the mice that are 16 months or even 20.5 months, you see this decrease in the proportion. But oddly enough, when you go through the fasting protocol, and specifically when you go through fasting, or these mice go through fasting, you see this sudden rebound of this ratio between the lymphoid and the myeloid cells. So they're the same, or similar at least, to the younger mice. So they completely flip from what you would normally compare, which would be to the older mice. So the old mice that fast end up having an immune profile that is more similar to the youngest level of mice, which is pretty incredible. Now, interestingly enough, when you do the refeed, you start to see this decrease of this ratio back down to the elderly mice, but you still see a significant difference between the similarly aged mice. So 20.5 months old, if they go through fasting, they're still going to have afterwards, after 7 days refeed, they're still going to have a substantial increase or change or positive change in their ratio of lymphoid to myeloid cells. Now, if that continues on, that's a question that really you have to ask yourself. If they had measured after a refeed of, let's say, 14 days or 21 days, would you still see that effect? I would probably argue that most likely it's starting to move back down to its normal state, which is where that white bar is the equal aged mice that are 20.5 months. Okay, ultimately, what can we get away from this particular paper, or this section of this paper in the second part of this content that I'm going to be releasing, or that I am releasing? Let's break it down into the three sections. Stem cells. What does fasting do for stem cells? Again, I would say that the literature is tending or leaning towards that that fasting has a regenerative impact, so it increases the population of cells and of course makes those cells also just a bit healthier. But I would caution and say that even though we have a little bit of data in humans, and it does look somewhat promising, because again, we just saw a trend, that doesn't necessarily mean that it would absolutely be the case if we ran a full study. But with that trend and with the mouse data, potentially there is at least an open door. We'd have to walk through it, but there is an open door for fasting to have an impact, a positive impact on our stem cells, the number of stem cells, and of course the health of those cells. Now, moving on to cancer, I think that the research was incredibly telling when it came to cancer. Not only did we see smaller tumors Not only did we see decreases in the number of tumors, the stage at which those tumors appeared, the deaths from tumors, from cancer, all decreased. All of those markers got better with fasting mimicking dieting. And again, you're not fasting all the time, you're just fasting for a period of the month, and then you're going to be repeating it, month after month, or every 2 months, or every 3 months. In humans, they did it a few times within a 3-month period, but they did not look at cancer markers for this particular paper. They only looked at the mice, so we only have the mouse data. So can we necessarily extrapolate that to humans? As usual, I'm going to say we have to be incredibly hesitant to do that, but because it is a mammalian model, at least it gives us, and because it's so unequivocal that there is an impact there, that fasting really does have a substantial impact, it would be really exciting to see that in future research, and just to see that in research in general. So I would say, based on the paper, that fasting does have an impact on cancer in mice. Now, with that said, let's go on to immunology. We definitely see that fasting has an impact on our immunology, on our immune system. Not only does it change the number of white blood cells, but it also changes the proportion to be better, and that's during the fasting phase. Primarily, we see decreases in the white blood cells, which might be indicative of something good, it might be indicative of something bad. We can't really say one way or another, but we do see this rebound back up to younger mice. So most likely, I would argue that because you would imagine that younger mice are going to be in a better state than older mice, you would imagine that fasting has a positive impact on your white blood cell count, and it does increase your white blood cell count to a robust level, back to kind of younger levels after the fasting period is over. During the fasting, we saw decreases in those white blood cell counts. Now, moving on from there, and that is, of course, all assuming that you do not have an infection, because if you go to the doctor and they check your white blood cell count and it is through the roof, that is not a good sign, because most likely that is something actually wrong with your body. But moving something from like a low physiological range to a high physiological range, that doesn't necessarily implicate your health. That could actually be a benefit. It all depends on context. And that is the second piece of content based off of this paper. We will see a third and a fourth piece of content covering this paper on far more topics like neurology. We're looking at lifespan, looking at health span, things of that nature. So without further ado, I'm going to end it here. I hope that you found this informative, and I hope that I have the pleasure of speaking with you in the next one. Have a good one, guys. See ya.